Hello listeners, Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, I have a few announcements to make. We are working on a new program geared towards transitioning from academia to industry. Powered by Dr. GPCR, our new program is called Life, Career, Job. The goal is to support you in your efforts to find your next job in industry, plan out your career trajectory, create a life, and support the lifestyle that works for you. If you'd like to be notified when this program becomes available, fill out our short survey at lifecareerjob.com. This new ecosystem will also include a new podcast that will be launching in September 2021. Our guests will be scientists, recruiters, and hiring managers, and everything in between. We want to help Ph.Ds find their ideal career path and inspire you with real-life stories. The second edition of the Dr. GPCR Summit will be held from September 13 to 19, 2021. We are planning a combination of live talks, pre-recorded talks, and live workshops. We have a great lineup of live talk speakers, including Dr. Brian Roth, Dr. Patrick Sexton, and Dr. Jürgen Weiss. Also, we are happy to announce that we'll have a live Q&A session with Dr. Bob Lefkowitz and Dr. Randy Hall to talk about GPCR's science and Dr. Lefkowitz's memoir. Visit drgpcr.com/summit today to see the full program. We're also launching our GPCR consulting services. You'll soon be able to find the profiles of our carefully selected consultants on our website at drgpcr.com/consulting. You'll be able to find the help you need for your company. Stay tuned. Are you subscribed to our YouTube channel? If not, please subscribe today. It's not only a great way to catch up on our recorded events, such as the Dr. GPCR Virtual Cafe, but it's also a great way to support Dr. GPCR. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR. I'm very excited today to have with me three guests, three Addgenes, Mike, Jason, and Nyla. Welcome.
Hey, nice to see you.
Thanks for having us.
I'm so happy that you were able to join us today, especially as a follow-up discussion to last week's presentation from Joanne about Addgene at the Dr. GPCR Virtual Cafe. And this is a great occasion for us to get to know you as scientists better, but get to know you as people who work at Addgene better. So let's start with some introductions, and I am going to pick Mike, who's at my left on the screen. And then, Mike, you get to pick who goes next.
Sounds like a plan. Yeah. Hello, my name is Mike Lemieux, and I serve as manager of Addgene's scientific support team. Nyla, take it away.
Hi. Yep. I'm Nyla. I'm also on the scientific support team here.
Hi, I'm Jason, and I am also on the scientific support team, and I also work with viral vectors and with the outreach team here at Addgene.
That's fantastic. So you're all in the support team, but you're also all scientists. You're also all PhDs.
Yep, we're all senior scientists.
Which is fantastic. And when you think about Addgene in general, you think about, going on a website, you find your plasmid and and there you go. And at some point, something gets in the mail and you have your plasmid. Can you tell us a little bit more about your backgrounds and then about your everyday roles at Addgene? And let's start with you, Nyla.
Sure. I got my PhD at the University of Pittsburgh in the GPCR field. My thesis was basically on cyclic AMP dynamics, looking at the thyroid stimulating hormone receptor. So gotta love those from GPCRs. And, while doing that, I used a lot of optogenetics, biosensors, and just all the other downstream, interesting tools that the GPCR field has. Went and did a postdoc at the University of Vermont, and it was mostly on cellular migration, but sort of had overlapped with a little bit of GPCR work.
Of course.
And then came over to Addgene, starting on as a senior scientist here. And I joined Addgene January 2020, so just before everything shut down. The world, not Addgene.
Well, yeah, before we pressed record, we were just talking about working from home. So I guess you barely got to see physically your coworkers, and then it got shut down.
Pretty much. But I get to see everyone virtually, so it works out.
I think so too. Someone told me once that we're not socially distant, we're physically distant, but we're socially closer than ever. So, Jason, tell us a little bit about yourself.
Yeah, hi. So I got my PhD in neuroscience from Ohio State University. And part of my thesis, I did receptor work with adrenergic receptors. So we were looking at how norepinephrine or noradrenaline, depending on how you want to call it, modulates synaptic transmission both presynaptically and postsynaptically. So that was a lot of fun to see. I did a lot of electrophysiology, so we got to see like in real time when these changes are actually happening in slice physiology. And so it was really interesting to see. I took a really non-traditional path in my education. So I finished my bachelor's, and then I worked in just kind of some state level work. And then I went back and got a master's, and I decided I wanted to go work in industry. So I worked for GlaxoSmithKline for a couple of years, and I really enjoyed that work. And then I decided that I didn't really have the kind of upward mobility that I wanted as a scientist working kind of at a master's level in a big pharma company. So I went back to school. I got both my master's and my PhD at Ohio State and finished my PhD. And then I did a couple of postdocs trying to look into different fields of academia. And I finally decided that I think my career path going forward would be better kind of outside of academia, but still having my hands a lot in science. And so Addgene produced, was a great opportunity because I get to stay really involved with the science. The questions that we get from the users of our viral vectors is really challenging. And so I'm constantly reading, I'm constantly learning, and it's a really great place, I think.
So it's a great intersection between, doing the science, but also having that human connection.
Absolutely.
That's fantastic. And Mike, how about you? Oh, I think Mike froze.
Hopefully he'll come in soon.
That's okay. We can wait for him to unfreeze. He looks like he's really paying attention and listening to us. It's a nice image to freeze on.
Honestly, once you get to know Mike, he needs no introduction. He's fantastic. So looks like his audio is coming in a little bit here.
Oh, there you go.
I'm so sorry for that technical difficulty. How much of that did you catch?
Well, not much, but you froze in a position that looked like you're really intently listening, so it was perfect. We were talking.
Start again.
Yes, please.
No problem. Yeah, and again, sorry for the technical difficulties in the Zoom world we're in right now, but I completed my PhD basically studying cellular motility. In the model organism Dictyostelium discoideum, which, if you've not heard of it, is a little soil amoeba. And the thing that I absolutely adore about it is that you just cannot kill these cells. Like, mammalian cells are super finicky and require the most stringent growth conditions, or they all die. You can literally grow Dictyostelium on your benchtop, feed it once a week, and it nothing kills it except direct sunlight. Go figure. So I did a lot of work on cell motility in the actin cytoskeleton, and I did not do any postdoc work, but like Jason, my path through my PhD was a bit non-traditional. I learned about midway through that the academic track probably was not going to be for me. I wasn't really up for scrapping for grant money and fighting for the associate professorship and tenure and all that. So I took it upon myself to find some industrial internships, and I completed one with Pfizer as part of their summer student worker program. Loved that experience. And it further reinforced the fact that I did not want to do bench work for the remainder of my career. And so it really kind of helped steer me in the direction that I ultimately ended up going. Started doing more networking, professional development, met the executive director of Addgene, Joanne Kamens, at a big ASCB meeting, which ultimately facilitated my path to the organization.
That's great. Thank you for that. All three of you touched on important points. One being that you all went through a PhD, you're all scientists, you're all savvy, but you also thought about what you liked and what you didn't like in your scientific everyday work. and based on that, knowing yourselves and knowing what those were, you really went and chose a career path that was tailored to your personality, to your lifestyle that you were looking for. And those things seem trivial, but they're actually very important. And I think a lot of PhDs and a lot of postdocs can appreciate the self discovery and figuring out what is it that you want to do. And I think that's a really important point to highlight, is that whenever you're a PhD or you're a postdoc in academia, there are other ways that you can work as a scientist. There are other ways that you can use your brain. You can still interact with people, be a scientist every day, other than going for a professorship, other than working in the lab.
I think that you hit on a really, really good point there. Going into my PhD program, for me, there were two career paths. There was academia and industry, and I didn't really understand much more about even what those two main paths entailed, let alone options that didn't necessarily fit into either one of those. And just being at Addgene, we place such a strong emphasis on professional development here and education here that I've learned more about what I can do professionally since I've come to Addgene than I learned in my entire school-based life beforehand. So
that's fantastic, and I like what you were saying, because so last week we had the live talk by Joanne, and she touched on a very important point as well, is that Addgene's website is not just an interface for scientists to go purchase or deposit plasmids, but there is a lot of effort put into education, and there are a lot of resources on Addgene's website, which I didn't know, to be honest, until Joanne pointed it out. So I think that's a nice way to tell the audience at this point, if they have a chance, just go on Addgene's website. Oh, we have someone joining us, so I'm going to let them in.
Sure.
On Addgene's website, and get to learn more about the different resources that Addgene has to offer. So here's my next question. Please go ahead.
Oh, I was just going to say, that's one of the most common things we get. And even for myself, I previously ordered plasmids. I saw some of their educational content, but I had no idea the wealth of resources they had, the details it goes into. And then of course with our scientific support team, the level of technical expertise that they offer has really been really cool. Very fantastic.
That's great. Please, that takes us actually to my next question. You had all different paths. You had master degrees, went to industry, came back and forth. Why did you pick Addgene? What makes Addgene different?
I can go ahead and start with that. I think one of the things that made it really stand out was every company has a mission statement, something that they're striving for, or something like this. Addgene, after interviewing everyone and talking to everyone, everyone was really authentically invested in that mission, and I think that's what really made it stand out. It's really it's been a great pioneer in sort of open science or reproducible science. If you've ever ordered plasmids from us, it's versus requesting it from a professor, you sort of notice the difference or the ease that goes into it. And you also have that guarantee of, if something goes wrong, you have someone you can write to who will, answer their email.
Yeah.
But yeah, that mission statement was just very authentically embraced by everyone here.
That's great. I did order plasmids from Addgene, and it was seamless to the point that I don't even remember anything other than I ordered them, they grew, I was able to clone whatever I needed into it, and my work was done. It was really, really seamless. So I think at that point, that was the whole point of putting the work into the efforts that you do every day.
Exactly the review we hope to hear. If it's seamless and not memorable, that's probably a good thing. But
I never left the review, but I think I'm going to go back and leave a review on those.
Thanks.
All right, I'll tag. Go ahead.
Go ahead, Nyla. Who are you going to tag?
I was just going to tag Jason. So
go for it.
Yeah, so actually, Joanne is what initially drew me towards Addgene. I had ordered plasmids when I was doing my graduate research work and trying to build an AAV vector, and that was great. But when I was doing a postdoc, Joanne came and gave a talk to our postdoc group, and just the way that she described her kind of philosophy on work and philosophy on careers and how you can kind of jump and change around, and how, at Addgene, that's something that they actually, strive to do, is to help people be their best selves, no matter what it is they're doing, even if, that career path takes them away from Addgene, which I also find was kind of shocking to hear from a CEO. I was like, well, if you want to go somewhere, okay, fine, we'll help you get there. So the environment is just really fantastic. And, the mission of trying to help scientists be open and to openly share resources and to provide, a context of help as much as possible. And that sense of being open is something that I think it's great for the entire community. And we say that we want that, but a lot of scientists don't actively go towards that. And so I think Addgene provides a great resource to do that.
Agreed. I was recording a podcast episode with, interestingly, an American scientist who moved to Europe. And usually it's the opposite. People come from Europe to moving to the US. But this person was mentioning that she took on a project about beta-arrestins, and a postdoc had worked on it previously. And after a couple of months of working, she decided, something is wrong. I'm going to resequence the plasmid. And there was a stop codon where there shouldn't be. And now that I'm talking to you, I wish that, at that time, Addgene would have had the plasmid, and then it would have been entirely sequenced, and the stop codon would have been at the right position.
Mm-hmm. I've had that happen to me too. Just a GFP, luckily it was a very easy marker, just got a random stop codon in the middle. So it happens.
It does. So Mike, what's your take on why you picked Addgene? You mentioned that you met Joanne at an ESCB meeting.
I did. Yeah. And her passion, I think, is what stood out to me initially. Just number one, having access to the executive director of an organization, I think, is something that stood out as well. I mean, how many organizations send their executive directors out into the world to communicate with students and postdocs, and so just meeting her face to face was impressionable. And her clear passion for what she did, for what Addgene did and stood for, really made a mark on me. And so I kept in touch with her, and the opportunity to potentially join the quality control team arose fortuitously right about around the same time I was looking to graduate. And that worked out. And I ended up joining Addgene in 2015 as a QC scientist. And I loved it, and quickly learned, just like Nyla and Jason, just how genuine of a group this really is. Being a mission-driven organization really does mean something. I mean, it means something externally, about the work that we do and why we do it. But it also means that internally, we value people. We value each and every employee at the company, and we really go above and beyond to help people get to where they really want to be. And we really, really try to make work fun. Shining example of that is we literally have a fun committee whose job it is to meet once a month to figure out how to make people happy at the organization. And that's just who we are, and I think beyond that, I mean, I could talk all day about the fun and the cultural aspects, but there's another aspect about Addgene that has kept me here for over 5 years. And that was the revelation that I don't have to be at a bench to do science, and to really, explore that natural interest in science that I've always had. I came to work for the company as a QC scientist, which meant I was at a computer doing science. And I learned that I actually loved analyzing DNA sequences and learning more about different technologies. Joining the Sci support team now means that I interact with researchers literally all over the world and help them accelerate their research without having to run the experiments myself, which for me is a great thing. And so beyond that, there's scientific communication, contributing to our educational resources, all things that are not in a wet lab that have motivated and driven me that I continue to be able to do. Yeah.
That's fantastic, which leads me to my next question. How do your days look like at Addgene? Anyone, and how do you interact with each other?
Yeah, so I've only started in the end of August of last year, so I've actually never met Mike and Nyla in person, or almost
So
anybody
true.
on the team. So my daily life is a little bit, it starts with kind of answering customer emails, trying to help them through some troubles that they may have. And then the middle part of my day is usually spent we're building, we have an AAV data hub. So adeno-associated viruses are an initiative that we started about 4 years ago.
Mm-hmm.
And one of the things that we want to do is share other people's experience with these, because they can be kind of tricky to work with. So we're trying to build out as much data information as we can and supply that to our customers and to whoever else wants to look at our website, so that they can use these tools more effectively, spend less time trying to troubleshoot things or trying to figure out which one's going to work best in their system, and get right to doing the hard science that they need to do.
That sounds exciting. And yet you're not at the bench during the day when you do this, which is super awesome.
Mm-hmm. Yeah, I'd say even though the majority of the day is answering emails, what goes into each email can be pretty unique. Sometimes it's a quick hey, I'm starting out a CRISPR experiment and I'm just looking for resources, or I read this but I don't understand it. Can you explain it to me? Offering some of that basic understanding to scientists who are starting out, versus really advanced questions like I'm looking to do this cloning experiment. Although we don't really offer cloning expertise, sometimes they're looking for maybe they aren't familiar with multi-cistronic vectors. So you're explaining on a much higher level to someone, with more advanced research, you should consider looking at these articles and these resources to get started. And then one of the other common questions, is I ordered something, I ran a digest and it doesn't look right. What's going on? So we actually get to, it's so nice. We get to, we have, other teams in the lab where we can click a button, design the digest experiment or sequencing reaction we want. Someone in the lab does it. They're absolutely fantastic, always working hard, and I trust their hands, I trust their eyes. They do really great work, and then I just get to look at the data. So I click a button, I get the data, and then I get to have the fun of analyzing it, figuring out what the problem is, and then figuring out how to troubleshoot it. So I get to do experiments, but I don't have to do the pipetting. So.
Absolutely, yeah. Similar to Jason and Nyla, a good amount of scientific support makes up a portion of my day. Like they explained, basically working with researchers using our products, anything from helping them to find resources in our repository that might be useful in their own studies, to explaining more about how a various resource might be used experimentally, to more rarely troubleshooting any issues they may have encountered in working with those reagents and quickly resolving them. So the human interaction is really nice. Beyond that, I and members of my team help with quality control. So every new deposit that comes into our repository, every single plasmid is validated in full by NGS, and we thoroughly analyze those results to make sure plasmids are what they're supposed to be before we make them available for distribution. Yeah. And we're almost up to 100,000 plasmids online, by the way. So Cool stat. And, moving away from the support in the QC, two more things that we do a lot of, scientific communication, I kind of touched on in my last response. We really contribute a lot to Addgene's educational resources, things like blogs, video protocols, helping scientists learn how to do various aspects of their work as efficiently and as well as possible is something that we're passionate about. And, personally, since I've taken on a management role, one thing I've really gained an appreciation for is just how many meetings managers are in and just how much you have to communicate with other teams at your organization. And so that is a significant portion of my day-to-day is cross-team communication and thinking about future projects or talking about projects we're already working on and how are they going? How can we improve things? So more strategic type thinking. Yeah.
Oh, that's amazing. What type of teams you communicate with? The way I see it, so you're about 100 people at Addgene. I see you all in different little circles, and each circle has a number of people in there. But within your, to your left and to your right, what circles do you usually communicate with?
Yeah, that's a great point. Our team's kind of unique, and one of the things I love about it is that we get to interact with so many teams. I mentioned the lab team earlier. We work with them for troubleshooting specific plasmids, and we also communicate with the depositors, like the PI who says, like, I want to distribute my plasmid with you guys, and coordinating, that arriving and getting to the lab, and if there are any issues with that plasmid while they're trying to transform it or something, if maybe there's something odd about it or it needs something unique, going back to the depositors and like talking with them about that. And Jason actually has a very unique role between a couple different teams. You want to take that?
Yeah, so I think I interact with almost everybody now, because I'm kind of like an intermediate between an outreach scientist and a support scientist. I interact with our viral production team regularly. I interact with our strategy teams in terms of like what viruses are we going to produce in the future, and kind of how do we strategize our production limitations with the demands for what viruses are out there. We recently started a new service for helping customers. So a lot of customers have like experimental questions on how to use their viruses. So we're now doing one-on-one Zoom calls to help people specifically with experiments. So it may go outside of what quality control that we normally do, but it's amazing to be able to interact with. I think next week I'll talk to somebody from my 7th different country since we launched it in February, which is just amazing. To be face to face with scientists from literally all over the world.
Yeah, and then the marketing and communication teams does a lot of the outreach and educational work that we were talking about. So those resources and sometimes helping them answer questions that come in on Twitter. And recently, like I was working with the sort of one of the outreach teams that works with distributors in other countries to help answer like scientific questions that way.
That's so fascinating. I think you are all three of you are giving us an inside look into how Addgene actually works. Because from the outside, it seems, as I said, my experience was seamless, and I didn't even stop to think about all the strategy and all the logistics that are behind the services that you offer.
Yeah. I suppose we also have our, of course, quality control team and curation team, and the curation team does a lot with there's a couple resource pages that like say I'm looking for a fluorescent protein and it's very generic. Sometimes a search engine is hard because if you type in GFP, you'll get a million things. But they have like very nice curated pages for like certain topics or certain pathways on our website too. So yeah, I think I got them all. Am I missing any, Mike?
For me, I don't think anybody's mentioned our legal team yet.
Oh, yep.
And, this was something new for me when I came to work for Addgene, is coming to appreciate the fact that there is a whole legal side to transferring materials, and plasmids in this case is the main material. And so, every deposit comes with legal paperwork. Every order and plasmid that we ship comes with its own material transfer agreement. And sometimes I have questions about that side of things, and being able to talk with lawyers on a weekly basis is kind of cool. It's something that I never got to do in graduate school.
It's like you're part of the series Suits. You have all your little lawyers.
And of course the actual customer support team that helps with like orders and some of the other like processing that way.
Wow, that's fantastic. So you're really able to communicate with so many different people at so many levels within the company, which is fantastic. You never feel isolated, although we've been socially distant for quite some time now.
Yes.
Very true.
So that gets me to my next question. You were mentioning helping scientists deposit their samples. Can one of you or each of you walk us through how does that work? Let's say I'm a scientist. I am a scientist, but I don't have any DNA samples to deposit, but I want to deposit a sample. What do I do? And how do you facilitate that?
Yeah, I can go ahead and take this. So, for example, in my PhD, I made some new construct. It was like an optogenetic protein fused to a luciferase. I'm still trying to convince my professor to deposit it with Addgene.
That's a shout out there.
carefully trying to see like, okay, so I might actually need to do this process. What do I need to do? So basically, we have an online document that you fill out all the information about the plasmid, the plasmid sequence, all the other like little pieces about it, and it gets sent to us. Once it gets to us, we have our legal and curation team check it to make sure that, for example, something like some luciferases have licensing agreements or are patented by certain companies. So some of those we can accept. Some of those we can accept with exceptions, and some of those we actually can't accept. So first it goes through that barrier of, are we allowed to distribute this to, nonprofit and like other academic institutions? Then the, tech transfer team works with sharing the material between the university. So like, even though the material is going to us, anytime we distribute it, the MTA is actually between the two organizations that, it's being transferred to. So that's an interesting thing there. So once that's all in, our shipping team, who we also interact with, sends you a nice little deposit kit saying, hey, this is what you need to do to prepare your sample. We accept, we'd like it as, DNA or sometimes bacterial streaked out on the agar plate. And you get a little kit, label it, barcode it with, prepaid shipping materials. It's completely free to deposit. We try to make it easy as possible. And it gets, shipped to us, where the lab team can then take it. We transform it in whichever, bacterial, we have a couple bacterial strains that we use, and grow it, make our glycerol stocks, make a backup glycerol stock, make a backup glycerol stock that's stored, in a like a super, liquid nitrogen freezer off-site, just in case anything ever happens. Like that's our, our super backup. And then our quality control team does next generation sequencing on it, which we've brought in-house recently, which is been an exciting initiative. We get the data, analyze it. Finally, if there are any problems, we go back to the depositor, ask them, hey, we saw this mutation. Did you know about this? Should we get a replacement? Is this fine? And sometimes it's, oh, I made a mistake typing my sequence, or, oh, that's terrible, let me send you a new one. And then finally, once that MTA paperwork's finished, the sequencing is fine, that goes online. You have the sequence displayed, all the information that you entered initially trying to deposit that plasmid, and now it's available. So
that's great. It's sounds like an easy peasy, simple enough for the scientists that deposit the sample, but there's a lot of work that goes on up until from the moment they contact you to the moment they actually the material goes up online. And yes, I do remember that Joanne had mentioned that you triple triple of everything. I love that, because you're right. Anything happens, you do have a backup, and a backup of a backup as well.
So yeah.
The other this is really cool. I'm looking at the questions we had pulled together, and I think we kind of explained or gone through the one of the questions I had for you is, how do you support the customers, which is a lot of email support, and then Jason, you mentioned that you have Zoom meetings as well with customers. Anything else extra that you do on an everyday basis, on a regular basis, to support customers that we haven't discussed yet?
Yeah, so we do offer a mix of what we've historically called technical and more currently scientific support. And as Nyla mentioned previously, there's also more general customer-based support, because as part of the plasmid acquisition process, one may or may not have a question of a scientific nature. So depending upon the nature of the question, we do have a main phone line that one can call, and a phone tree where you can choose the appropriate team that you may wish to speak to. So we offer that option as well. Or if you prefer email as a customer, you can shoot a general email to a very easy to remember address, help@addgene.org, and we will triage that email and get you over to the most appropriate team who can quickly assist you. Jason, I definitely want to shout him out for piloting kind of a new way of offering science support, the Zoom-based office hours. This is new for Addgene, and not something that we've tried in the past. And seems to be working well. And correct me if I'm wrong, Jason, but so far so good with that.
Yeah, we've had, we just started this service in February, and I think we've had appointments every week since we launched it, and it's been kind of a soft launch. We haven't advertised other than we do a Twitter feed. We tweet once a week, and then I let people know about it through some of our help desks as we're trying to get understand the machinations between how we contact customers and keep track of things. But it's been really exciting because, working with viruses in the brain tends to be tricky. And one virus may work great with neuron A, but it may not work well with neuron B. And we get a lot of questions about this. And so, just being able to lend the experience that I've had working with these viruses and talk to customers and just kind of help them as best that we can through that, it's one, really rewarding. And two, I think the customers find it really like a really nice way to kind of get over some humps at times.
It sounds like a really great initiative. And especially, do you think, well, my guess the answer is yes to this, but I'm still going to ask it. Do you think it's something that is going to stay on as an initiative, as a staple of Addgene's scientific support in the future once we go back at some point to the office?
I hope so. Yeah, I think it will. I mean, whenever something like this gets launched, you never know exactly how it's going to be received. Initially, when I first put out some emails, I didn't really get any response. I launched it at a SFN meeting, Society for Neuroscience meeting, and we weren't getting initial responses. But then as soon as the calendar actually got up and people started hearing about a little bit more about it, it just kept rolling. So I hope so.
I'm pretty sure it will. I think it's a very unique approach. It's a very personalized approach. Mm-hmm. And what a cool way to stay connected to your customers. Because at the end of the day, you may help someone on day one, but then a couple of years down the line, they've produced materials that they'll be happy to give back to Addgene for distribution, based on the discussion that you had. So I think it's a nice way to give and also receive.
Yeah, I think building relationships is the most rewarding part of that. It's just great to talk to people and hear about their science.
Yeah, especially that, which I also didn't know, that Addgene is international and you have distribution centers all around the world. And that also, feeds into your ability to talk to people from all around the world without having to travel.
Yeah, I think we just shipped to our 106th country this past week.
Wow, that's phenomenal. So as well, but the audience also knows that Addgene is a not-for-profit company. What does it mean to you to work for a nonprofit?
For me, I really like that, very often things become a balance of time or money. Here, I can actually just make quality as a focus. And I think that's so important for science. As you know, even if one little thing is off, your entire experiment can give you, something different. So the fact that we can really focus on quality without having to worry about the burden of, a bottom line or something, makes it really, it makes it very rewarding. It, I don't know, I just really like it, and it's where my own values lie. So I appreciate it.
Absolutely, yeah. I second those feelings, and I think the nonprofit aspect has at least two major perks for me, and one we've talked about previously, in that being mission driven really, I think, is core to our culture and results in a company culture that is very employee and personal or person focused, and so that matters a lot, because ultimately it means we're happy at work. and that means so much, coming from, particularly in my case, a training program where I wasn't very happy at work a lot of the time. And so being able to work for an organization now where happiness is the norm means a whole lot to me. And, the second aspect of this is the accessibility aspect, where core to our mission is improving access to useful research tools. And being a nonprofit means we provide research tools at much lower costs than one might be able to find them elsewhere. And so by providing them at that much lower cost, That's how we improve accessibility and make sure that more people can do the science they need to do.
With the materials that, with the best starting materials that know are controlled for.
Yep.
Yeah, I think it also allows us, like, one of our main missions is to be open, and as a nonprofit, we don't develop a lot of the vectors that we don't develop any of the vectors that we distribute. They come from academic labs who have worked hard on them, and we're able to be completely open with everything that we know about them, which I think is really a great resource, both as a company and somebody to work for a company, but also just for the scientific community in general. It just really, you want to know something about one of our plasmids? Here's the sequence. You want to see what QC we did? Here it is. Take it. We try to be as open as we possibly can.
Yeah, and I think that definitely contributes to democratizing science.
Mm-hmm.
And reproducibility. We talked about this with Joanne, and that's a big issue. And I think, other than democratizing science, Addgene also helps in making sure that reproducibility is something that we're aware of, but also contributes towards allowing that to happen with great starting materials. So this is a Dr. GPCR podcast, so I have to ask a GPCR focused question. Can you tell us please a little bit about the products that Addgene has to offer to GPCR scientists?
I think Jason and I can definitely weigh in on this. Obviously, there are too many to name because every single GPCR, there's just a ton of plasmids out there. And so, of course, there are, GPCRs with tags, truncated versions of GPCRs, all throughout, like, these individual plasmids that, GPCR labs have donated to us and wanted to share. But, like, there are also some really big, interesting collections that have come through. Like, for example, I'll just highlight the Brian Roth lab. They've been very, very forward and, openly sharing their materials. And so there's one like the Presto Tango GPCR collection, which is basically like a beta recruitment assay linked with a luciferase reporter. And it's been made so that you can screen like the druggable human GPCR ohm. And then they also have like the TruePath collection, which is a series of Brett biosensors. It's an assay for G proteins. And so, like, it helps you identify the subunits. So it just kind of gets to that, like, yes, there are GPCRs, but there are also all these unique tools related to the GPCR field. And that's one of the things I really liked about studying GPCRs is all of the tools and things that have come that are linked to it. For example, I mentioned I was doing optogenetics and chemogenetics. So we have entire pages of biosensors, optogenetics, chemogenetics, that don't have GPCR in the title, but are all related to the field.
Yeah. And you also, I went through the different plasmids that you have, and they're not only receptors, but they're also G proteins, beta arrestins, anything you can think of. There's other labs also, but I think Brian Roth's lab is one of the big contributors to Addgene. And it's interesting you mentioned it because I think just a couple of days ago, Brian had posted on Twitter that they've reached over, I think, 10,000 a huge number of people who actually requested their plasmids. And I think the number that stood out, and I can't remember what it stood for, was 666. But 666, and then I thought it was, that's the thing that struck me. Yeah.
Maybe number of reagents. I'm not sure.
I think so. I think it was the number. I think it was the number of reagents that they had submitted to Addgene, and the 10,000 was the number of times any of those reagents were distributed. That
could be it. I have to confirm, but that could be it. Yeah.
Yeah. And I'll actually touch on that a little bit later if you don't mind, but I'll let Jason answer.
Yeah, the things that I'm most interested in, as a neuroscientist, are some of these GPCR-based biosensors that we have available, both in plasmid and in viral vector form. And so these are vectors that sense different molecules like neurotransmitters such as glutamate or norepinephrine or dopamine, and they're basically a GPCR that's fused with a GFP that has a conformational change, so you get increases in fluorescence based upon the concentration of your ligand, which I think are just absolutely amazing. And I wish when I was in grad school these things were available, because it would have really sped up my dissertation.
Yeah. So we have a ton of tools, but for GPCR scientists who want to deposit, since you mentioned, the number of plasmids that the Ross lab was distributing for a bit. So when you distribute with us, or start depositing with Addgene, we also send you monthly emails saying how many times your plasmid was deposited. And so while, in the beginning, that's, certainly motivational, be like, yeah, the stuff that I'm making in my small lab is, being spread across the world. It has like a list of countries. You get bragging rights, right? But what we've seen, especially in the last couple of years, is like, some researchers have started adding that number into their grants, showing that the work that they're doing is having an impact out. And this is a nice quantifiable way to show that, like, I'm contributing to science, and this is why I deserve a grant, because I'm helping the larger community, and it's not just my one paper, my one lab.
That's fantastic. You touched on a very important and interesting point, and it's something I think it's really valuable to put out there. And it also, as you said, it shows how many people are not only reading your paper, but they're actually willing and able to test and use your materials as a scientist and validate your research. And it touches on reproducibility, but also on popularity, let's say, of your work, which is fantastic. That's a nice way actually to put it in a grant application and say, 10,000 people or 10,000 requests for 666 plasmids.
Yeah.
That is fantastic. So you're all from different backgrounds. You love it at Addgene, but I have to ask, where do you see yourselves in 5 years? And it's not it sounds like an interview question, but not necessarily as where will you be working in 5 years? But you mentioned, Mike, that Addgene puts a lot of effort into developing people and their careers and get them evolved during their time at Addgene. Where would you like to be in 5 years?
Yeah, I think that this is such a difficult question, and it
is.
It's it really is, because it really forces you to think, what makes me tick, what makes me happy, and honestly, sometimes I don't know what I'm going to be doing in 2 months, let alone 5 years. But if I had to think about it and answer it, as you said, Addgene puts such a strong emphasis on professional development. And one of the things that I've enjoyed so much about my role in the last 18 months is developing my manager's toolbox. I love working with people. I've always loved working with people. I love how being involved with a team at an organization like Addgene just forces me to communicate and work with so many people with such diverse backgrounds. And I've loved kind of organizing a team. I love helping people do their work to the best of their ability and manage their own projects. So in 5 years, I hope to be a more seasoned, effective manager. I would love the opportunity to work with more and more diverse groups of individuals. Maybe an expanding team is one thing that I can foresee. With Addgene's distribution numbers on the rise and business going very well, thank God, I would love to see our team grow, and I would love to continue to help develop those team members. Yeah.
Thank you. That's fantastic.
Yeah. So I'll follow up. I might have a bit of a cop-out answer, because I'd love to still be working at Addgene. There's a lot of cool initiatives that are kind of on the horizon that I think are really, I think they're big. I think they're really important, and I think they'll make some very impactful changes. But if not at Addgene, what do I like about my current job that I want to continue doing? I really love helping people. It's just been a genuine driving factor throughout everything, and I'm very curious. The reason I became a scientist is because that driving curiosity. So a place where I can obviously take advantage of both. It doesn't necessarily need to be at the bench, which is what I've learned. I've very much enjoyed, being able to do it through this way. I actually really like the sort of technical support area because it has that bridge of quality control versus like working with other researchers and, talking with them, helping them troubleshoot. So it's definitely I definitely want to be in a very similar area to where I am now.
Mm-hmm. Super. Jason, you're the last but not least.
Yeah. So one of the things that I found is something that always has driven me with science and in my career is that one, I love figuring out how to solve problems. And I love talking to people about those types of things. And so I think in 5 years, either here at Addgene, I'd like to find new and innovative ways to kind of talk to more people, help more people with their science and their experiments. I don't want to do any more of my own bench work, but I would love to be able to just continue to help other people do that and maybe find an innovative way to communicate better or to help people in a way that I don't know of at this point in time. But definitely I have to be talking with more people constantly.
That's fantastic. And I think in one way, this is what we're doing. We're trying to, I'm introducing you to the audience, but at the same time, we're talking about Addgene, and we're also talking about career trajectories. And that takes me to my next question. What would be your advice for scientists who aspire to make science more reproducible and also aspire to find their path?
Get out of your lab if you are a student or postdoc. To do anything in your power to zoom out a little bit and pull yourself out of the work that you're currently doing and talk to people. I mean, you can even start with the people you're surrounded by every day, because something that I came to learn later on in my own graduate career was that even though we're all, kind of at the same organization doing graduate work or what have you, we're not all going to be doing the same thing a few years down the road. We all have different interests, and we're all using different strategies to realize those interests. We just don't talk about it. It's very rare that you have like a discussion about careers or career paths with your immediate coworkers, and that needs to be more normalized, I think. So look for any opportunity you can to open a discussion like that, even with the people that are really close to you every day, and then expand from there. Go to conferences, networking events, get online, social media communities like Twitter can be great for this. Just talk and meet as many people as you can. It's probably the most important thing in my view.
Agreed. I think it's very important to go out there and get out of your bubble, because I think when you're working in the lab, you can very easily get lost in the details and focus on what's not working and trying to solve it. But I think taking a break and talking to people and getting to learn what others are doing is beneficial, not only for you, but for the other person or the other people you meet. And at the same time, it opens your horizon, but it also gives your brain time to solve any problems that you might have left at the bench.
Yeah, for me in grad school, I was very lucky that shout out to the University of Pittsburgh for having a really good a lot of career development series, and a lot of it was just sort of organized and run by the student group associations. Nice. So I'd say take a look at your university. Chances are there are already things in place with alternative careers in science seminars or things like that. And if there's not, make one. You'd be surprised how receptive people are to a graduate student request. Hey, would you mind giving a seminar at our university and telling us about your career? A lot of people really like to talk about their career. So setting up things like that gives you very it's not just like you're going to a networking event and seeing who's there, and one person could talk to a bunch of different people today but have no lasting connections. Whereas if you organized a seminar, invited a speaker, you're getting much more direct networking experience. You're also bringing in exactly the sort of things that you want to learn about more, and it also helps the community that you're working within.
That's fantastic advice, Nyla. I love that.
We actually had Joanne come for our Women in Medicine and Science Association. And that was a very very obviously worked out pretty well because I became, that was my introduction to Addgene from beyond the screen. So
yeah, that's phenomenal. Jason, you look like you wanted to add something.
Yeah, I mean, one of the things I think about that I never did as a grad student or a postdoc is, I was always so focused on my science and like, what's the next thing I need to solve, and the minutia that you get into. It's stopping to take a minute to think about, well, what is it that I like about this? And what is it that I can do that I'm going to continue to like about this? And if it's an academic path, well, what happens when you're actually not at the bench anymore and you're an actual professor? How much work are you doing? How much grant writing are you doing? How much paper writing are you doing? Yeah. And really evaluating, is that the lifestyle you want? Do you want to interact with more people? Do you want to have, really understanding yourself, I think. And then getting out there, like Michael said, talk to a bunch of different people. Go to like your tech transfer office and talk about what goes on behind the science, or go to, a different department like the legal department and say, hey, what do you guys do with biotech or PhDs or patent offices? There's a lot of things that, if the way that the academic system is set up now, academia, and the professors who work with know academia. And the only thing that they can say is that you could go into industry and you're just going to keep doing the same pigeonhole thing time and time again, which isn't necessarily true, because, they don't have that experience yet. And people in industry have that industry experience and academic experience, but there's also this whole swath of information or swaths of career paths that are a little bit more obscure or not known about, but can also be like incredibly rewarding, like science writing or doing science communications or science policy. There's just no structured way to learn about those things. So get out there and talk to more people.
Agreed. You could even, please go ahead.
Oh, yeah, sorry. I was going to add to that and just say, I think it's normal to think about this in the context of, graduate students and postdocs mostly. But I think a really important point here, and again, something I've come to learn at Addgene, is one can do this at any point in one's career, right? It's never too late to talk to more people or to expand one's network and start doing things like this. So it doesn't matter at what point you're at, it's okay to start.
Exactly. And I think it's not, you touch on a very important point. I think all of you do. It's okay to start, even if you feel like, oh, it's late. You don't tell yourself I should have started yesterday. There's no time like the present. And it's not a finite thing that you have to do. I think it's think of it as a marathon. It's not a sprint, because you never know who you're going to meet. And I think Nyla and Mike, you both met Joanne, and that's the connection that you formed to Addgene.
Yeah, all three actually.
Yeah. See, all of you, and that's how you came to know Addgene. And that's an important point, that you always have to be open to meeting new people. You always have to be people facing. You never know. It's a small world. And an introduction, a 15-minute chat could lead to your next opportunity, or it could lead to a connection that then at some point could lead to the next opportunity, or you can even get to help someone. So I think it's a marathon, basically.
Yeah, I like to think of it as a hike.
Yep.
And all of a sudden there'll be a new trail you didn't even know existed. And you just take a little deviation, and maybe it brings you back to the same trail, and maybe it won't. You just don't know.
I love that. I love that image. A hike in the forest, and then you never know where you're going to end up. Super. And on this note, I think I want to thank all three of you for being here today. I had a lot of fun chatting with you, and people can find most of you on LinkedIn. Actually, all three of you on LinkedIn, and also on Twitter. I know Jason and Mike, you're on Twitter. I invite everyone in the audience, if you want to reach out to Addgene at addgene.org, or to Jason and Nyla and Michael, just look them up. Our website will have your contact information and your link to your LinkedIn profiles. And with this, I'd like to thank all three of you for joining us today.
Yeah, thanks for reaching out. This is great.
Awesome. Thanks so much. Of course. Talk to you soon. Bye, everyone.
Thank you for joining us for this Dr. GPCR podcast episode. I'd like to thank our guest, as well as our team members, Attila Forrest, Shivani Sajdev, Alexa Duran, and Ines Pinero. We look forward to seeing you live at the Dr. GPCR Summit in September. Don't forget to visit drgpcr.com/summit. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us testimonials at drgpcr.com/testimonials. Another way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.